I would like to welcome those who have just joined the session. We'll be starting with today's session in more 5 minutes. Thank you for joining. And in the meanwhile, we begin with the session. You can introduce yourself in the chat box and tell us about your educational or research background and also your research interest. Thank you. We are looking forward to hear from you. Hello, everyone. I would like to welcome you all for the very first session of Bioinformatics for Precision Medicine program. This program is designed to introduce students and researchers to the field of bioinformatics applications in precision medicine. In this program, we will explore how the various omics data types generated in these studies can be analyzed and integrated to study the basic biology associated with viruses and their hosts, cancer onset and development and outcomes. This online program will include weekly sessions with an instructor, online tracking and review of students' progress, access to all resources and materials online for self-paced learning, access to expert guidance and problem solving, educational and research pipelines, and domain data analysis on our big data server. In this session, we will discuss about overview of precision medicine program, current applications of bioinformatics in precision medicine, and much more. So to begin with, what is precision medicine? Precision medicine is an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. This approach will allow doctors and researchers to predict more accurately which treatment and prevention strategy for a particular disease will work in which group of people. It is in contrast to a one-size-fits-all approach, in which disease treatment and prevention strategies are developed for the average person with less consideration for the differences between individuals. After decades of research, we are poised to enter a new era of medical practice where detailed genetic and other molecular information about a patient's cancer is routinely used to deploy effective patient-specific remedies to treat it. We are about to enter the era of precision medicine, where biology is now seen as a field of data science. Biology is rapidly acquiring the characters of data science. Billions of data on genes, proteins, and other molecules are compiled in large files and systematically studied. Analyzing this data will lead to more knowledge and understanding about living organisms, including human health, biotechnology, crops, and livestock. This program will give an overview of bioinformatics for students, biologists, and clinicians. The influx of data, associated methodologies, and data-driven research makes bioinformatics for life sciences as prominent as the emergence of computers for the tech industry. The goal of the omics logic programs is to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life science and data science. The Omics Logic portal gives students options for analyzing a given dataset, code-dependent paths or code-independent paths. These datasets and associated publications can be used in several ways. You can learn bioinformatics without any coding at all. For that, in each project, we explain the analysis pipelines on the user-friendly T-Bioinfo platform for big data analysis. You can also learn the scripts behind many of the steps, learning to wrangle, clean, visualize, and analyze data in popular languages like R and Python in order to create your own analysis. Finally, you can hear about the projects and their research applications in our mentor-guided programs where experts share their experience and support research projects developed by you. After using the T-Bioinfo platform to visualize the pipelines and practice analyzing, students are then able to take their theoretical knowledge, to remember and understand levels of blooms, and apply that in a project. They apply, analyze, and evaluate level of blooms. Then check their comprehension with levels and badges, certification of completion, and test. Similarly, faculty should be able to assign the Omics Logic portal to their students with the understanding that it will give them holistic and comprehensive knowledge of bioinformatics, beginning with bytes and molecules and on to designing their own question-based project. After completing this course, you will know how to do the following: read and understand bioinformatics literature, perform basic NGS data analysis processing steps, analyze data and form hypotheses, prepare data for machine learning, cross-check signals with gene databases, know what type of data is required to answer scientific questions, create your own bioinformatics analysis, present your results in an organized way. All these skills are essential in bioinformatics and some can be translated to other fields. So let's start out by signing up on the learn.omics logic portal. This educational portal will help you get started on your bioinformatics journey. Thank you, Jasri, for putting the link in the chat box. If you have not yet signed up on the Omics Logic portal, you can take this time to create an account for yourself. Our team is here if you face any of the technical issues, so we'll be here to assist you with that. You can either create an account for yourself by putting in the details that are required, or simply sign in by using any one of your social media accounts. That would include a Google account, Facebook, or your Apple ID, or Orchid, or Twitter. Thank you, Dr. Fatima. Those who have already signed up can maybe let us know in the chat box by sending in a 1 or yes, and others can take this time to create an account for themselves. Meanwhile, I'll go ahead. So those who have created their accounts already are requested to complete their profile. A completed profile will have a profile picture, a full name with appropriate capitalization. You should add the links to your social media handles and also add a brief bio about yourself that will consist of educational background and research interests. The mentors can view your progress under the Activity tab. The courses you are currently completing will appear under the Courses tab. And finally, after the completion of your course, the certificates will appear under the Certificates tab. So for different parts of the session, we will leverage omics logic resources that you can use to learn about precision medicine. Asynchronous coursework with theoretical material, quizzes, and practical assignments. Our cloud-based analytical server where you can analyze large genomic data and visualize results. The Omics Logic Code Playground where you can practice coding in R and Python, as well as a repository of project examples. And these methods will include hands-on exercises on the T-Bioinfo platform, which provides a simple and intuitive user interface for processing and analysis and machine learning methods for multi-omics data. Throughout the course, the users will be given the directions to access the T-Bioinfo platform for processing and analysis of RNA-seq data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. Different stages of analysis are performed in different sections of this multi-omics platform. And behind many of these methods are scripts which are written in popular coding languages such as R and Python. And these are also explained in the tutorials that you will have the access to as a part of this program. So from visualization of genomic data to exploratory analysis and hand hypothesis testing, we have it all covered. After you register, you will be redirected to the Courses tab. Here you can search for any course title, use the tags to find series of resources around the topic, or choose one of the collections down below. Here we will look at the Getting Started collection. Once you complete a course, you will gain 500 points. For running a pipeline, you will get 100 points, and for updating your profile, you will get another 100 points. Finally, participation in group activities will give you 10 points. And after achieving the learning outcomes, you would have learned some of the major best practices in the field of bioinformatics. You will be exposed to R and Python, which are the two most powerful programming languages. Bioinformaticians will need to understand how to use t-tests, regression, and other tests, analyzing and managing data in order to communicate important information is another best practice in bioinformatics. Pipeline development and utilization is another common practice. Ultimately, completing the precision medicine program will help you enter the field of bioinformatics since you will learn several best practices. Now, I would like to ask Ojasvi to take over the stage and also play for us the recording by our CEO, Mr. Ilya Brodsky, for today's session. Ojasvi, stage is all yours.
Precision Medicine. Welcome to the Omics Logic Bioinformatics for Precision Medicine program. Precision medicine is changing the way we understand, diagnose, and treat major life-threatening diseases. This transformation is driven in part by computational technologies, as well as high-throughput molecular data that is being collected from patients, animal models, and large-scale cell line experiments. In the program, we will explore how the various omics data types generated in these studies can be analyzed and integrated into the clinical process to understand the basic biology associated with communicable or infectious, as well as non-communicable or chronic diseases, including diagnostic strategies and treatment selection, development, and repurposing. So in the first session, we'll review again the main topics of the program, introduce each one in greater detail, and provide you with a guide how to use associated resources to continue this journey. After this session, you should have a good sense of bioinformatic applications in precision medicine. Completing the session video and associated resources will give you a great grasp on how data analysis skills and new technologies like next-generation sequencing are used today in this field. You will also see various examples that you can use to focus on oncology, neurodegenerative, and infectious diseases. In this session, we will learn about clinical and molecular data in the context of risk profiling, diagnosis, and therapeutic decisions. We will also go through various next-generation sequencing data types such as genomics, transcriptomics, and metagenomics. Finally, we will learn about the process of how discovery in the lab leads to real-life applications, or how insights from bioinformatics analysis can be translated to the clinic. To understand clinical decisions where bioinformatics is used for precision medicine, we will look at various data sources such as basic information about patients, or clinical data like age, sex, ethnicity, and their environment, data that describes disease history and diagnosis, treatments, and comorbidities, and molecular profiling that allows us to perform genotyping based on SNPs in the DNA, identify biomarkers, or look at microbiome composition. Clinical data typically includes data such as demographics, labs, and medical history. The list of collected variables can vary significantly based on each patient. In most cases, this data can come from a variety of sources like electronic health records, medication, insurance, claims, and family history. For the most part, it is divided into patient information, as well as describes their interactions or encounters with the medical provider. Recently, the sources have increasingly digitized and integrated, leading to an expanded number of variables with various relevancy for each case. Some studies are starting to incorporate many of the clinical sources of information with molecular profiling, environmental factors, as well as mobile health and even social media. While some of the data types are more accurate than others, these all have been used to study disease. For example, in the current pandemic, all of these have been used to track disease spread and identify potential at-risk individuals or groups. Pharmaceutical companies are referring to this as real-world data, or data that can be used to evaluate responses to treatment and improve selection of patient candidates for clinical trials. For many diseases, molecular data is increasingly important and relies on complex bioinformatics analyses, like gene expression analyses, commonly found SNPs, metabolism information, and an array of various biomarkers of disease and treatment efficacy. These molecular patterns in association with clinical data can help refine strategies for diagnostics and therapy selection. New discoveries in molecular data lead to a better understanding of complex diseases at the subcellular level and help identify novel drug targets or opportunities for drug repurposing. For the clinician, advanced molecular profiling reveals the underlying biological drivers of disease and can help explain disease symptoms using genomics, proteomics, transcriptomics, epigenomics, and even metagenomics. These molecular markers affect how a patient is diagnosed and lead to a number of improvements: risk prediction or risk stratification, prognosis of outcomes for treatment modalities, such as individual drugs or combinations of drugs, selection of target therapeutics for patients with complex diseases, and continuous monitoring of disease changes over time. One of the great examples of how such treatment has affected cancer is the example of breast cancer. So we see that from 1971 to 2011, the rate of survival has increased from about 50% to over 85%. One of the reasons was because studying cancer, Herceptin was developed as a targeted therapeutic. It's a monoclonal antibody that has been used to treat HER2-positive breast cancer patients since the 1990s. It works by targeting the HER2 receptor, preventing cancer growth. Some breast cancers express too much HER2 protein, triggering the cells to multiply very rapidly. Today, this trend with targeted treatment has increased. Other monoclonal antibodies, such as pertuzumab or Perjeta, have been developed. The targeted therapy has gotten even more advanced. TDM1, Kadicla, an antibody-drug combination, has been approved for use in the treatment of HER2-positive breast cancers. Antibody-drug combinations work like a smart bomb, delivering chemotherapy directly to cancer cells by attaching to their HER2-positive receptors. Targeted therapies have transformed the approach to breast cancers and offer an opportunity to cure even later-stage breast cancers in certain patients. Now, a lot of this is done using molecular profiling. One such test, called Oncotype DX, looks at the expression of 21 different genes in a patient's primary tumor. A tumor's gene expression pattern shows whether or not it will be responsive to chemotherapy or whether endocrine therapy alone, such as tamoxifen, could be a better choice. A recent study showed that patients scoring on the low to mid-risk range could safely skip chemotherapy completely, which would lead to avoiding the hair loss, neuropathy, weight loss, and other side effects that often come with chemotherapy. But this approach has further developed, and today, molecular markers of disease have led to a complete reclassification of breast cancers into several different molecular subtypes. These subtypes continue to evolve, leading to a better treatment of this complex disease, which is the fifth leading cause of cancer-related deaths annually, with more than 265,000 people diagnosed with it in the United States alone every year. Other multi-omics studies help change the way that we can understand other cancers, such as liver cancer or hepatocellular carcinoma. In the 2017 publication in Cell Reports, the Cancer Genome Atlas Research Network published their analysis of integrated multi-omics data that led to a much better understanding of liver cancer risk, such as infection with hepatitis B and C, as well as other factors like alcohol consumption, ethnicity, and other comorbidities. Such comprehensive studies continue to yield results even today. More data is being collected and analyzed by bioinformaticians, leading to an even greater precision and making it possible to improve the way treatment is delivered or even find new treatments that are less harmful and more effective for certain patients. The role of next-generation sequencing in medicine has changed dramatically over the past decade. To understand why and how, let's look at several methods of molecular profiling, explore the process of second-generation high-throughput sequencing, also known as next-generation sequencing or NGS, and see how such data can be analyzed to discover specific clinically relevant data points for a medical procedure. The growth in high-throughput data is largely driven by cheaper, faster, and more efficient sequencing called next-generation sequencing or simply NGS. From about 100 million in 2001, which is the cost of what it took to sequence the full human genome, to approximately 3 million by 2008, and by 2017, the cost of a sequenced genome was close to $1,000. Just recently, Ultimate Genomics announced its goal to make the full genome sequence available at a revolutionary cost of $100. This places whole genome sequencing at a comparable cost to most tests, like the X-ray. Recall that it's been over 70 years since the structure and function of DNA was discovered. 50 years have been spent since that time sequencing the human genomes. In 2015, the 1000 Genome Project brought a significant number of whole genomes sequenced and available to the research community. 100,000 whole genomes were already available by 2018. And today, there's an ever-increasing number of datasets that are available to help researchers and clinicians understand subcellular processes and the DNA. Omics data, including such that is provided by next-generation sequencing, includes a variety of types. Phenomics refers to high-throughput imaging or characterization of phenotype. Genomics allows us to identify mutations in the genome. So what is the observed sequence compared to the reference? Epigenomics allows us to study epigenetic variation, or variation that is not dependent on the actual sequence of the genome. This includes such methods as DNA methylation. Transcriptomics allows us to understand gene expression, or how much of a certain gene is being expressed in a cell or a tissue. This is done using a variety of platforms. Platforms are instruments that produce digital omics data. So those can be divided into two types: short read sequencers and long read sequencers. And they can be used to sequence either targeted, amplicon, or whole genome or untargeted DNA and RNA sequencings. In most cases, for clinical use, we will refer to either whole exome or targeted panel sequencing. Because in most cases, whole genome sequencing just carries too much information to be clinically relevant. The number of annotated SNPs is much lower than the total number of positions in the human genome, which is over 3 billion long. Genomic variation is typically calculated by the proportion of known SNPs present in a particular gene. And this can range from genotyping, where we group patients by their genotype or some special features in their genome. And in some cases, we are going to be looking for pathogenic SNPs. SNPs are single nucleotide polymorphisms, or a single nucleotide position that has been identified as relatively prevalent in the population, or over 1% of individuals. For example, BRCA1 and BRCA2 testing is extensively used to identify women that are at risk of developing breast or ovarian cancer. In fact, 44% of women who inherit a harmful BRCA1 genetic mutation will develop ovarian cancer by the age of 80. To summarize, omics data can be used to understand what is happening within the tissue or a single cell. And typically, those can include a variety of DNA changes, gene expression changes, epigenetic changes, as well as microbiome changes in the individual. When combined with clinical or phenotypic data, some of that information can be clinically relevant. That means that it will be used at different stages of identifying risk, developing a precise diagnosis, or even selecting treatment that is most appropriate and effective for the individual. And that is why next-generation sequencing data is central to precision medicine. Most bioinformatic analysis.
All right, so let's just pause for a moment and let's see if the participants have any queries related to the topics covered so far, or regarding the session or about the program. If so, please put them in the chat box below and we have them be happy to answer your queries. So we are waiting. So if you have any queries, please put them in the chat box and we'll be happy to answer. All right. So in the upcoming topics, we will be discussing about the process of how discovery in the lab leads to real-life applications and how insights from bioinformatics are used in the clinic. All right, so we have a query from Mukesh. Is this helpful in machine learning data science? So one of our mentor will be able to answer that.
Yeah, hello Mukesh. Yes, this will be helpful in setting up machine learning analysis. So for machine learning analysis, you will need a structured data. So performing a genomics data analysis or analysis from the FASTQ files or based on the sequence data is one of the way where you can achieve or where you can convert the unstructured digital data into structured digital data. Once the data is structured, or in another way, once the data is in the format of a table arranged as rows and columns, then you can use that data to variety of different machine learning models or different advanced statistical methods. So this will definitely help in data science methods using machine learning tools.
Thank you, Dr. Raghav. Does the participants have any further queries? Okay, we have one question. Will the session recordings be uploaded? My work timing coincides with class schedule.
So yes, the session recordings will be uploaded and would be made available to the participants of the program. And the time for the sessions would be the same, that is 10 a.m. CST and 8:30 p.m. IST. I hope that answers your question. Thank you.
All right.
And for the next question, yes, it is already replacing Sanger sequencing. Right? Next generation sequencing, high throughput sequencing methods are the most prevalent, and they are most sought after for genomics and transcriptomic studies, as it's been explained in these videos. So Sanger sequencing right now are the technology of the past. They are not high throughput sequencing. They are low throughput sequencing. And we have another question. What is the role of artificial intelligence in next generation sequencing? I mean, artificial intelligence doesn't have a specific role in the sequencing process itself. But as I mentioned, once the sequencing has been done, you are going to create lots and lots of digital data. Wherever you have the availability of a large volume of data, you can expect artificial intelligence to play a significant role in those areas, like finance, for example, like Internet of Things, and like astronomy, right? Astrophysics. So all of these fields are creating huge volumes of data, and you can see artificial intelligence are playing significant role in those areas. Likewise, in bioinformatics also, we will actually see a significant role from artificial intelligence to make sense or make relations or understand trends in the huge volume of data that we are going to generate or we are generating as such. What's the difference between second generation sequencing and third generation sequencing? I think, Mohammed, you can take this from these theories from Google also. It depends on the type of sequencing being performed. So all sequencing makes the aim of the sequencing is very simple: to convert what is a biological sample into a digital data. And there are different ways of achieving it, and each with each and every development that is being that has been in the process for these sequencing methods, there are slight increase in the signal to noise ratio for different ones. So if you want specific answers between second generation sequencing and third generation sequencing, for example, for bioinformatics examination, you can consult Wikipedia or Google, or even Illumina website is a very good source for these types of queries. Yes, and we have some more questions. Any relationship between single cell analysis and metabolomics, transcriptomics, and genomics? Daniel, yes, single cell analysis is mostly preferred for RNA-seq data, right? In order to understand or in order to characterize the heterogeneity that one will actually expect or one will actually find out in any sample. So bulk RNA-seq or bulk transcriptomics analysis is a way of characterizing samples by averaging out the expression patterns present in those samples. And single-cell RNA-seq or single-cell transcriptomics methods or processes are a way of trying to individualize those cells based on their expression pattern. So we will obviously find different applications for bulk RNA-seq or bulk transcriptomics analysis and single-cell RNA-seq. And there is another question by Sushmita. There is always a conflict about data analysis of omics data, as we have lots of data generated where the study objects sample individuals are less. How to deal with the problem? Should we add more replicates technically? If you can approach the problem of lower number of observations or lower number of samples from the data generation point of view, that is the best way to tackle this challenge. This is a challenge, and in some cases, this is a tremendous problem. And there are different methodologies that you can adapt your machine learning models or your machine learning codes or your different advanced statistical analysis, so that the impact of the lower number of samples remains very low and it does not affect your end result or your interpretation. So there are ways to get around these problems and issues. What is the prospectus and scope of genomic data analysis in terms of patient care and diagnosis? We are actually seeing the change right now, isn't that, Dr. Avinash? So for most of the cancer genomics or for most of the cancer patients, one of the sought out methods to characterize the cancer is to send the sample for transcriptomics or whole exome or whole genome analysis, right? So we not only are reliant on histological characterization of the samples, for example, we are also reliant, or we also want to perform genomic data characterization or omics data characterization of the samples, so that we can get a more, let's say, a perspective that is going to add to one more dimension of whatever has been available from histological laboratory, right? Genomics data or omics data analysis gives you the molecular perspective of the samples. And what the laboratories are producing, it gives you a different perspective of the samples. So together they can actually increase, or they can actually improve the diagnostics accuracy and prognostics accuracy. It is already seen, right? In developed countries, it's part of the regimen, and in developing countries like India and other countries, it is picking up. Sure.
Thank you so much, Dr. Raghav. So if the participants have any other queries, please put them in the chat box. And I would like to request Ojasvi to continue playing the session.
Analyses are going to be considered as preclinical. That means it is based on data generated in lab experiments or retrospective studies. To explore these, let's look at examples of cell lines derived from patient tumors in oncology, animal models of human disease, and explore how these can be used alongside databases on various specific diseases, for example, TCGA in oncology. Then we will discuss how such insights can be validated and what considerations they bring to biobanking and clinical trials. Next, technologies and experiments are already affecting research, hospitals, and are used by pharmaceutical industry to design specific treatments and adapt them to the needs of individual patients. A lot of times, not all of these experiments are possible on real patients. Instead, we can select tumors in different types of patients, produce those tumors as immortalized cell lines, and use those cell lines to understand the basic biology. Unfortunately, that biology cannot be fully studied just using cell lines, because cell lines don't have all of the elements of the system or the whole body that actually functions as a whole. And so in many cases, in oncology, instead of just using cell lines, there are also tumors that could be extracted in full from a patient, grafted into an animal model, and then studied in terms of their interaction with the rest of the body. Ultimately, such experiments can provide better insights into biological factors that are relevant for patients, and different types of tests could be first performed on these models before we actually develop a clinical trial where such treatments or diagnostic procedures are used on actual patients. This is also changing the way that we apply big data analysis techniques or bioinformatics in real-world clinical needs. So typical process is to collect, integrate, and analyze big data coming from a variety of sources and ultimately change the way that this can be used in a big data approach for precision medicine. We can see this oftentimes used when large-scale data is being collected on a particular disease. A good example is the Cancer Genome Atlas. The Cancer Genome Atlas is a work performed over several decades to collect samples with multi-omic data on patients and use their clinical phenotypes to find important associations. The cancer genome data is available through the Genome Data Commons portal that allows us to find multi-omics datasets with clinical information about each sample, which gives a much better understanding of how cancer variation is diagnosed and treated and how successful those treatments can be. These large-scale datasets are continuing to be collected, and today we have the All of Us dataset as well. The All of Us research program, which is led by the National Institutes of Health, is building one of the largest biomedical data resources of its kind. The All of Us Research Hub stores health data from a diverse group of participants from across the United States. Researchers can access All of Us data and tools to conduct studies to help improve our understanding of human health. Now, it also provides us with a much better understanding of what we call healthy, or the variation of healthy individuals that can be used as a reference when we study subgroups of patients or patients that are of different ages, sexes, ethnic backgrounds, and socioeconomic status. In addition, there are projects that are aimed at characterizing the healthy condition and preclinical variation. One such example is the NIH Human Microbiome Project that allows us to look at different types of microbiome perturbation that is associated with different types of diseases. These include depression, autism spectrum disorder, cardiovascular disease, asthma, arthritis, cancer, and diabetes. By understanding the microbiome diversity, composition, and changes associated with different types of phenotypes, we can start seeing the impact of the microbiome on diagnostics, but also treatment, because patients with different microbiome can metabolize treatment at different rates. Now that we have reviewed the rationale for bioinformatics and NGS in biomedical research in the clinic, let's review case studies of clinical research with real data. These include oncology, neurodegenerative diseases like Alzheimer's disease, and infectious diseases like COVID-19, malaria, or Ebola. We can look at how viral genomics and evolution can help us understand the spread of a disease and identify variants of concern in pathogens or genetic predisposition of some populations to be more susceptible to acquire disease and potentially have worse outcomes. We can look at immune responses, where we characterize gene expression and study biomarkers or genes that are associated with a particular response. And sometimes we can look at microbiome and see how microbiome plays a role in disease progression, drug metabolism, and also predisposes certain people to different clinical risks. On the Omics Logic portal, you will find a number of case studies and curated datasets around these different topics. Some of them are based on full analysis of datasets that are already published. Simply by following the steps described in these case studies, you will be able to make important discoveries that today are being used in the clinic. Some of them have to do with drug metabolism, identification of somatic mutations in tumor development, inherited or germline mutations like the sickle cell anemia, rare genetic diseases, as well as drug resistance associated with different types of antibiotics and pathogens. Oftentimes, just by reading the abstract, you get a good grasp of how bioinformaticians can use such data to model precision medicine. For example, on this project, modeling precision medicine of breast cancer, you will find how researchers use machine learning to study transcriptomic data and identify subgroups of individuals that are going to respond differently to different treatments. In the study, researchers use multi-omics data integration to look at different types of molecular profiles, integrate them, and compare them to reference databases like the TCGA to understand response to treatment to drugs that are either already used in the clinic or being studied in clinical trials. To continue learning, we invite you to explore these associated resources for the lecture. We will go over the coursework, the case studies, and other examples from literature. To benefit from today's lecture, you can select one of these to refresh your memory of basic terminology, learn more about the various techniques, or practice what we have learned with curated datasets on the topics that we covered in today's session. The contents of the session correspond with practical lessons that you can complete independently that are available on the Omics Logic Learn portal. With the full course, specific lessons that are of relevance to today's session include Lesson 2, Bioinformatics in Healthcare from Course 1, Introduction to Bioinformatics. In this lesson, you will get an overview of examples from healthcare area where bioinformatics can be applied. You will also see examples and videos of medical professionals discussing bioinformatics and precision medicine applications. Another good course to look at is Bytes and Molecules. This course is a great place for those who was confused by a lot of the terms that we used in today's lecture. Anyone that would like to review the basic terminology and get a good overview of topics that we cover in this course should look at this course in full. But specifically, we looked at this lesson 6, called Introduction to Genomics. If you are interested in genomics, you can also look at these additional lessons in the Course 3 course called Genomics. In Lesson 2, you will see how you can work with DNA sequences in R, and in Lesson 3, you will get an introduction to large-scale genomics. The full course contains a lot more information that you can choose to explore independently. Some of these will be covered in the future lectures, but do not worry about completing them for now. At the end, you will also find examples that I already mentioned. These examples include such projects like Ebola, immune response changes in cancer, modeling cancer precision medicine, or tuberculosis infection and treatment finding drug resistance variants. So let's recap what we have learned so far. First, we learned how combining clinical and molecular data helps reveal underlying factors contributing to the disease, which further helps us provide a precise diagnosis and effective treatment to the patient. Further, we learned about next-generation sequencing technologies for genomic, transcriptomic, and metagenomic data that provide important insights for precision medicine applications. We also discussed the role of bioinformatics in taking research results from the lab to the clinic to benefit patient treatment and diagnosis. Finally, we introduced a set of case studies in precision medicine, from inherited sickle cell anemia to drug resistance in Mycobacterium tuberculosis. In the next session, Session 2 on genomic data analysis, we will go one step further to better understand what is genomic data, how do you perform variant calling analysis, what is the difference between germline and somatic mutations, and how to determine a variant's clinical relevance. At the end of Session 2, you will have a thorough understanding of genomic data, variant calling analysis, as well as specific techniques for clinical annotation of variants in cancer.
Thank you, Ojasvi, for playing the session. And we have few more queries in the chat box. So first we have Divya is asking, would this include the R program for the analysis?
Divya, the associated resources also includes a specific lesson that does this analysis or part of the analysis with R as a tool. But most of the heavy analysis like a pipeline, running the pipeline is actually taken care by running it in the backend, which is the Bioinfo server, big data bioinformatics server. You need a high performance computing to work with the FASTQ files and then produce structured data. And then from these variants that are being called, which has to be, this variant calling pipeline has to be running the Bioinfo server. And from these variants, we can perform analysis with R to annotate them or to compare them, not as. So those are actually included in the associated resources. And yeah, some of them will be covered in the future lectures. So we also have occupational opportunities and with certificates for these courses. And yeah, can Sonalika actually talk about what are the certificates that will be provided after completing these courses?
Yes, I'll take that question up. I would be just talking about the registration part now, and also how you can go through these resources. So if we have no technical question left, I would now like to take this session ahead. All right. So I'm here on the Omics Logic Learn portal. Ojasvi had shared the link earlier in the chat box. I'll share it once again. If you have signed up, great. If you have not yet signed up, please take the minutes and do sign up. Having an account on the portal is absolutely free of cost, and you can proceed further by registering for the program to get the access to the resources that we were discussing about today. So all you need to do is just click on the login button, and it will allow you to create an account for yourself with several options. Yes, Divya, I'm just coming to that point where I'll be also discussing about the scholarship opportunities that you can avail, right? Okay, so meanwhile it's loading, and I would like to take you all to the program page. So here is the program page, and I wanted to discuss with you all the syllabus of the precision medicine program that we'll be going through in the next sessions, and also how to register yourself if you have not done so till now. This was the first session where we thought of inviting all the interested participants of the program so that you know how the sessions would be conducted. And because it was an introductory session today, we did not want to overwhelm you, so we kept it short and light. And this is the program page. I've shared the link in the chat box with all of you. When you scroll down, you can find the sessions schedule. So we have listed on which all dates. Okay, I think there's a system error over here. It should be July 14. Okay, so we have listed here all the topics that we would be covering for each and every session on like all the topics here we have listed. So today we discussed about introduction to bioinformatics or precision medicine. Next, we are going to talk about the advancements in precision medicine. And then we'll talk about genomic data analysis. Next, we'll discuss oncology data resources that would include raw data and public databases. After that, we are going to talk about the transcriptomic data analysis aspect for precision medicine. And then we'll talk about health, wellness, and ethical concerns. Next, we'll discuss, in the next session, we'll discuss about phylogenetic analysis and multiple sequence alignment, and then talk about host response to infections. We'll also discuss about precision medicine for neuroscience, and finally, metagenomic data analysis. So we have covered a whole range of topics here for various omics data analysis in the field of precision medicine. And here we have listed the topics that would be covered in each and every session. Now, a point to be noted here is that all the program participants will be able to access the session recordings here on the program page itself. We'll upload the session recording here on the program page once the session is over. And you can see the recording here under each and every session. Also, each and every session of the program will be supplemented with some coursework, associated online resources that you will be supposed to go through and complete. Once you go through the recording and also complete the associated resources, next you're supposed to click on the complete session button, and you have to do the same for each and every session. This way you will be able to get your certificate. So I'll just very briefly take you to the profile and certification tab as well and answer the questions related to certification and how do we get it, all the resources that are covered, etc. Before I do that, I would just very shortly like to discuss the registration steps. So we have 3 different subscription types that the interested user can pick from. One is for 45 days, another one is for 60 days, and the third one is for 90 days. Now, obviously, as the days are mentioned, you will get the access to the program, the resources that includes the Omics Logic Learn portal, all the courses out there, and also the Bioinfo server hands-on analysis platform. You'll get the access to all of these resources for 45 days if you pick the first option. And also we have tried to mention the other resources that you will get the access to. And if you pick the access for 60 days, then you will also get to work on your research proposal, further work on a research project after you are done with your training for the program. And the license type that you will get on the Bioinfo server would be that of an educational license. And you'll be allowed to also meet the experts in one-on-one sessions. And next we have the 90 days of the subscription level, where you will also work on a research project alongside your training part. So once your training is complete, you will work on a research project. And to work on a research project, you will have a research license on the Bioinfo server, where you'll be able to extract data from various big data repositories and then upload that data after preparing it on the Bioinfo server and do your own analysis, get your results, and that way you will be able to work on your own independent research project under mentor guidance. So that is the difference between these three different types of subscription levels that are possible for you to become a part of the program with. And I would now like to share the link to the scholarship form. So those who are interested to avail a scholarship on this program, please fill out this form. I will also put the link in the chat box. Please fill out the form on the scholarship page here. Our team will review your application and then get back to you on this. Once you have filled the form, please let us know. You can also email us at marketing@pint.bio or marketing@omicslogic.com for any further questions. Nilaksh, I'm not sure if that was the case. The sales team would be able to help you with that. Maybe you might have used a scholarship coupon code. That's why the amount for you might be less. All right. So that was it. Now going back to the page where if anyone has any questions regarding registration that we just discussed or scholarship form, please put it in the chat so that we can discuss that now. Yes, Jean, the prices for all the summer training programs are the same. CG Code Girl, could you please first of all tell us your full name in the chat box, and also this was the session for the registered participants as well, and we'll be having the next session only for the registered participants on June 16th, that is day after tomorrow. All right. Divya, I've answered your question for the possibility of getting scholarships. You can fill out the form. I'll also send the link to you once again so that you have it handy. Yes, eligible for the scholarships, Dr. Fatima. It's not specific to Indian citizens. It's for everyone around the world. I'm sending the link once again in the chat box. You can fill the form. Mukesh, it seems you're interested to enroll for data science or biomedical data program. We'll send you the details over an email. Please share your email ID with us in the chat itself. So for Mohammed, I'll just open our Bioinfo server to show you the platform. In the initial slides that I went through, I did discuss about the Bioinfo server, where you can deal with your own data or data from big data repositories and run various analysis pipelines for these different domains of transcriptomics, genomics, metagenomics, epigenomics, or data science as well, be it machine learning and supervised analysis, even single cell transcriptomics. So there's a huge possibility of data analysis on the Bioinfo platform. Once you register for the program, you will be going through a lot of resources and also practice hands-on pipelines on the server, and then get an understanding, a clear-cut understanding of how these pipelines work, how do you get the data, how do you download your output files, and then finally interpret biological insights. Okay, so Sparsh has shared the details in the chat itself. Thank you, Sparsh. And you can reach out to us at marketing@omicslogic.com and also contact us on our Indian number, which is active on WhatsApp as well. All right, so that was it for today's